Hi everybody, it's Dina again, and I'm going to finish up chapter 2 and talk a little bit about meiosis. I've also recorded another small video that explains it as well from something called study.com. But anyway, meiosis, it's really going to be good for you to sort of get your head around some of these terms, especially with chromosomes, because it does get a little confusing. But the big point is what I want to talk about most here, and that is in the process of a meiosis, it's very specialized cell division in which instead of having an exact copy of the amount of DNA and chromosome material in one cell, like starting with one cell and you duplicate it and then divide it out to be equal to the first one. So the mother and daughter cells have the same amount of chromosomes. In this case, in meiosis, you want a reduction of the amount of genetic material by 1/2. And that's very important for these to go into sex cells, the gametes, or the as they're called, whether they're male or female. And we want what's called a haploid number in there, or half the original number. And that's important because upon fertilization, you'll restore that original diploid number. It'll be different though, you'll have a different combination of things, which is really cool. And all of this is about maintaining genetic continuity from generation to generation, so that there's the same amount of information and there's variation. And the way variation will come across is there's a lot of things that we'll talk about even in the next chapter, chapter 3, but we'll talk about crossing over today. And that is basically you have homologous chromosomes that are duplicated during this process, and during that duplication, the one from your mom has not been duplicated, so you have 2 from your mom, you have 2 from your dad, and they'll do what's called, they'll line up again, but the homologs will cross over. Material from one will maybe join the material from the other, and they'll sort of form this union that's brief to kind of hold them together, and that gives it's an exchange of genes, so it makes even more combinations of genetic expression possible. So again, there's 2 cell divisions in meiosis that are required to reach that haploid number. It all starts with doubling, and then we have what we call it's meiosis 1 and meiosis 2, and each one of those divisions of meiosis have the typical phases of prophase, metaphase, anaphase, and telophase. But again, this goal is twofold, and this is the main thing to remember. This is the big picture thing, is to get half the chromosome material into these gametes, or the egg and sperm cell, and then also to recombine things, to mix things up to get a lot of genetic variety. And that's going to be a fundamental concept through the study of genetics, is how do we get this variety? We get it through a lot of ways, mutation and segregation, but crossing over is one way. All right, so some terms that I'm going to try to help. This gets a little confusing, and I'm going to encourage you to, after you listen to me, to go form your own drawings and opinions. But there's these terms that are confusing: chromosome and DNA and chromatids. And so basically, a chromosome is consists of DNA. That's a double helix, and it's bearing a linear sequence of genes. And we sort of looked at that picture in the first lecture. What happens is we coil it and recoil it around proteins called histones, and that resulting complex is called a chromatin. And then during cell division, when we replicate that, so it's almost like chromosome and chromatin are the same, except whether or not they're bounded with a connection with the centromere. And so when we've made this identical copy of DNA from a chromosome, when we connect it to its homolog at the center, we call that a chromatid. We call them sister chromatids. And so if it's before DNA replication is formed, it's just, there's some typos in there. I got to clean that up, but it's formed by one chromatid. And then after, it's after replication, it would be 2 chromatids. So anyway, it's worth getting it straight in your head. Sorry about the typos on that side. All right, let's see if I can go on. So this is sort of what it looks like. Here's a chromosome before replication. Now we're going to replicate it, and we're going to call these 2 parts of that replicated chromosome, they're sister chromatids. 2 chromatids or sister chromatids is often used. And, they sort of use this term as a chromosome and this as a chromosome. But it's, in this case, it's really been replicated, and it has 2 duplicated chromatids held together by a centromere. Hope that helps. So basically, prophase 1 of the first mitotic division is where the diploid cell will duplicate. It has already sort of done this. It's duplicated genetic material will kind of line up, except in this case, it's very similar to mitotic prophase, except these chromosomes pair up. And so you have almost a kind of a doubling of the doubling, and you get this, instead of being held together by a centromere, you get it like this, where there's a centromere holding them together. It's more like this, and there's some crossing over that occurs that helps ensure, and crossing over is an exchange. It kind of forms a little bond, and it holds this thing together. But there eventually will be centromeres, and they're going to be present, but they'll be at the they'll still be at the equator. But there's also this holding together of a synapsis, or a joined area. This gives rise to what's called, instead of having a pair of sister chromatids, now we have chromatids. We have two pair of sister chromatids, and it's this intertwining of these chromatids. We call that a chiasma, and or a tetrad, because there's 4 in this case. And again, that's all to ensure some recombination of genetic material. And then you'll get some of the normal stuff with the envelope breaking down, and these centromeres of the tetrad will attach to spindle fibers. And so look at some pictures in your book, and there will be a video to follow this as well to help explain that a little bit more. You know, they're really short at this phase, metaphase 1, and again, the other, you can read some of this about the point of contact. Again, this point of contact between these tetras are intended to ensure that they keep contacting, have the chance to recombine and cross over. Spindle fibers will eventually move them to the metaphase plate, and then that alignment of the tetras on the metaphase plate is also random, and how they segregate is random. We'll talk more about that in the next chapter, but that again leads to genetic variety of what's going in the egg and sperm. All right, anaphase 1, we've talked about this. It's the same sort of thing. This is when they're going to move to the poles, and this is where we call it disjunction, when half of the tetrad, which is then going to be called a dyad, moves randomly to the opposite pole. If there's non-disjunction, there might be more going one way than it should, and less being left behind. And so you have an uneven distribution of chromosomal material. But at the end of this phase, the number of dyads equal a haploid number of chromosomes that are present at the poles. You're going to have telophase 1 here, where you're going to have, and each cell is going to be haploid, and you might be thinking, how could that be? But hopefully, it's basically referring to a state where we've reduced with the number. You'll see in the video a little bit more explanation about that. But at this point, we're holding them together with the centromere, and they're not going to replicate. They already consist of these sister chromatid pairs. And so the second division will occur, and this is going to be very similar to mitotic division. But at the end of the day, we're going to get 4 haploid gametes that are going to result from this event, which is it's called the second meiotic division, but it's really an act of mitosis. And so if you look at it this way in a picture, this is meiosis 1. This is the group that's been duplicated, doubled, and there's crossing over, hopefully, and the pair of chromosomes condense. In metaphase 1, they line up. And then there's going to be anaphase and telophase. Then the homologs are going to be held together into the haploid daughter cells. So we have, they're haploid because we've got half the genetic material here, and they stay haploid here, and they stay haploid here. They're just divided. It's still half of the genetic material that is in here. And so it's a little bit weird to kind of do it this way, but try to get your head around it a little bit. But the main thing here to keep in mind from a big picture thing is that there's a lot of genetic variety that ends up being produced because of how cells divide. And you can see, based on the crossing over, you can see how this cell is pretty much like the original, more or less, with the colors. But you can see these have combined colors, meaning that that's a lot of genetic variety. So depending on which egg is going to get fertilized by which sperm, there's a whole lot of possibilities. And so somebody once said, from a two-paired couple, the chances of the different combinations of genes represent about 64 trillion. So it's pretty cool stuff. And that's very, very important. Variety is good in genetics. Keep that in mind always. All right, let's look quickly at how these male and female gametes are produced, these haploid cells. One of maleness is called spermatogenesis. We're going to make that. And the female gamete is called oogenesis. And it's basically the making of these sex cells, the gametes that are going to be haploid. Spermatogenesis takes place in the testes, and oogenesis obviously takes place in the ovaries. They're very complicated steps and processes, and we're not going to get into them to the nth degree, but we'll do them here in brief summary. Spermatogonium is a term, and sorry about this, this picture needs to go away. Whoops. Anyway, up here it says spermatogonium, and this is oogonium. Those are basically stem cells that will differentiate into spermatocytes and oocytes, and there's divisions that occur. This is all basically describing meiosis. Sperm is pretty straightforward in the meiotic process, but the oogonium and the oocytes, there's development of what's called a follicle when we do oogenesis on a female side, and there's these things. Only one of these follicles will actually mature and get released into from the ovary into the fallopian tube for fertilization. And the interesting thing about this whole stuff with female gamete production is really the sperm make a bazillion. Men make a lot of sperm all day long, every minute of every day. For women, the number of eggs was predetermined at birth, and so what happens is there's a certain number of eggs produced, but they don't develop into follicles or they don't divide. It's arrested at one phase of meiosis, mitosis, and it doesn't start dividing again into these secondary follicles until after puberty, and that's when you start to either fertilize or ovulate them out through menstruation. So they either ovulate out to the fallopian tubes, or they get ovulated out as part of the menstrual cycle. And that there's still arrested development, though, because it's still not haploid at this point. The oocytes are technically diploid. And throughout this process, there are polar bodies that are given off because they have to sort of dump genetic divisions because only one is going to mature. And so, again, there's two arrested states for making an egg, and by the time it's actually haploid, it's really instantaneously only haploid right before fertilization. It needs chemicals from the sperm cell, so it needs to be basically ovulated and released into the fallopian tubes. It's still diploid. It's really odd. I don't know why this design occurs, but it gets into the ovum, and the ovum gets into the fallopian tubes. It's waiting to meet a sperm. If it meets one, it's chemical signaling from that sperm that will trigger the last division to make that haploid, and then it fertilizes it right away. It's really bizarre. But anyway, spermatogenesis, let's back up a little bit. It's when these undifferentiated spermatogonia germ cells, I would call them stem cells, become a primary spermicide. This is all taking place in the testes in an area called the seminiferous vesicles. But there's just the basic idea here is that it produces motile spermatozoa or sperm, and it uses the processes of meiosis too. Oogenesis we've already talked about, and the reason, one of the other interesting things about this, if you looked at a sperm, and if you want to Google this and get a picture of the sperm, it's basically three parts. I went back a little bit, sorry. It's got a head that contains half the genetic material, because the other half went to a different sperm. It's got to carry just half. The next part of the sperm is like a neck area, and it's loaded with mitochondria. And that's it. As far as organelles, that's the only organelle really that you find in a cell is the nucleated genetic material and mitochondria to give it energy to propel that flagella as it needs to move. It contains no cytoplasm. So when you get over to the egg is huge and big, and it is has a lot of cytoplasm and organelles in it, like other mitochondria and different Golgi apparatus and things like that. So genetically speaking, only female mitochondria is a unique cellular organism. They even think it maybe evolved from bacteria. It has its own DNA. It's really kind of bizarre. But that DNA all comes from your mother's side because the sperm has no cytoplasm. It has no mitochondria to pass along. So you can do genetic studies and ancestral studies from the maternal side by looking at maternal DNA that's found in mitochondria. So anyway, that's why the bulk of cytoplasm is very important there, and that also serves to help give nourishment to a developing embryo as well. All right, again, these polar bodies are created at various steps of the way, but again, that secondary division is really completed right almost instantaneously with fertilization. It's really bizarre. All right, again, to summarize here, meiosis is critical to get sexual reproduction of diploid organisms. In the sexual reproduction of diploid organisms, we want to get to the haploid number, and some other plants and animals do things a little bit differently, but anytime there's an exchange of genetic information called sexual reproduction, there's gametes involved. And so another big theory of this is the crossing over that occurs during the first meiotic prophase. It further reshuffles these gene alleles between homologous maternal and paternal chromosomes, and they segregate and sort independently into gametes. This results in a great amount of genetic variability, and we'll look at segregation and independent assortment in the next chapter a little bit. So I think that's it. There's some pictures here of just different electron microscopy pictures. You can see this kind of led to the theory of what I want to end on, a couple of things here, and then I'll get going. Again, it led to this folded fiber model theory. It's not that important, but basically it led us to understand how we can do about 5,000 folds of compaction with the length of DNA to get it into the chromatin fiber. I mean, it's really phenomenally, I don't know if you believe in a creator or science, but either way, it's really, really cool. If you stretch the DNA in one cell all the way out, you have about 2 meters long of just DNA from one cell. If you stretched out the DNA in all of your cells and put it together, it'd be twice the diameter of the solar system. So that's pretty cool. One other thing I'll leave you on, if you really, really want to see a good documentary on egg and sperm, and mainly sperm, it's about an hour and it's fabulous. You will love it, trust me. It's called The Great Sperm Race, something like that. I'll put a link to it on Canvas, but it's worth watching. They scale up what it's like to be a sperm as it tries to get to an egg, and they scale it up using humans. And so you'll see the different stages that the sperm really goes through a lot to try to meet the egg, and it's fabulous, fabulous documentary. I will post a link to it, or you can search by it. I think it's called The Great Sperm Race. So that's it. All right, thanks a bunch.